In this video, we are going to look at the histology or the microscopic features of epithelial tissue and learn how to identify these tissue when provided under a microscope. This is a syllabus of your first year anatomy practicals, and in this video you will learn the tricks that you need to know in order to identify these epithelial tissues. First, let's look at a brief description about epithelial tissue. So, epithelial tissue, it's of two types. Simple epithelium, where cells are arranged in a single layer, and compound epithelium, where cells are arranged in multiple layers. Now, while observing under a microscope, you may not be able to see the cells properly. In that case, you can look at the nucleus of the cells. If the nucleus, they are arranged in a single layer, that means the cells around it is also arranged in single layer. So it's simple epithelium. But then if nucleus, they are arranged in multiple layers, then obviously the cells are arranged in multiple layers as well. So that's your compound epithelium. Simple epithelium, on the basis of the shape of the cell, it's of three types. There's your simple squamous epithelium, where you will find flat cells with flat nucleus arranged in a single layer. So you need to know the feature that it contains flat nucleus. That is exactly what you are going to observe under a microscope, the presence of flat nucleus. Second is simple cuboidal, where you will find cube-shaped cells arranged in a single layer, and these cube-shaped cells, they contain round nucleus. Again, remember the type of nucleus, round nucleus. And finally, simple columnar epithelium, where you will find these tall pillar-like cells containing oval nucleus towards the base of the cell. So they're simple squamous with flat nucleus, simple cuboidal with round nucleus, and simple columnar with oval shaped nucleus. Then there's your pseudostratified ciliated simple columnar epithelium. Now throughout your specimen, if the cilia is present, it's more likely that it is pseudostratified epithelium, because that's the only specimen that is going to be provided to you in your practicals. So this is what you need to see. First, if there is cilia or not. Second, observe the nucleus if the cells are not properly visible. The nucleus are going to be at two different or multiple levels, and this is due to the difference in size of the cells. This right here is your pseudostratified ciliated simple columnar epithelium. Okay, one last time. For simple squamous, flat cells with flat nucleus in single layer. Simple cuboidal, cube-like cells with round nucleus in a single layer. Simple columnar epithelium, oval nucleus at the base of the pillar-like cell arranged in a single layer. Pseudostratified ciliated simple columnar epithelium, presence of cilia and nucleus at different levels. Then there's your compound epithelium, where you will find cells in multiple layers, or the nucleus of cells in multiple layers. Under this, you have stratified squamous epithelium. So you'll find nucleus in multiple layers, and the topmost cells, I repeat, the topmost cells are going to be flat. Okay. On the basis of presence or absence of keratin outside the epithelium, it can either be keratinized or non-keratinized. I'll tell you how to distinguish between the two of them under microscope. Then there's your stratified cuboidal. Again, you will find nucleus in multiple layers, and the cells present at the topmost level, they are cube-like or cuboidal. Then finally, there's your transitional epithelium. In transitional epithelium, cells are arranged in multiple layers. However, these cells, they don't have a fixed shape like squamous, cuboidal or columnar. But what you can see is that the cells at the base, they are round. The cells in the middle, they are pear-shaped. And the cells at the top, they are dome-shaped. So that's your transitional epithelium. Now let's look at the microscopic features of each of them. What you have here is a section of Bowman's capsule. It could also be alveoli. Either way, whether you are provided with a section of Bowman's capsule or alveoli, you'll be able to observe one thing, and that is spaces. As soon as you observe the spaces, you can guess that this has to be simple squamous epithelium. For assurance, you can observe that there are these flat nucleus. There are these flat nucleus, and these flat nucleus, they are present in a single layer. So this has to be simple squamous epithelium. So two things: the presence of spaces marks it as being either Bowman's capsule or alveoli, and you know that in Bowman's capsule and alveoli you have simple squamous epithelium. Microscopically, you can see that there are flat cells, and these flat cells are arranged in a single layer. You may not be able to see the cells. Look at the nucleus. You will find flat nucleus, and these flat nucleus are arranged in a single layer. This is simple squamous epithelium. Second, so you can see these follicles. Surrounding these follicles are cells, and these cells, they contain round nucleus, arranged in a single layer. So this is obviously simple cuboidal epithelium. Up third. So we are done with simple squamous with flat nucleus, simple cuboidal with round nucleus. Now you have these pillar-like elongated cells containing oval nucleus present at the base of the cells, and you can see that these nucleus, they are arranged in a single layer. So this is your simple columnar epithelium. So we are done with simple squamous with flat nucleus, simple cuboidal with round nucleus, and simple columnar with oval shaped nucleus. Next, as soon as you observe cilia, there's a high probability it is pseudostratified. But then look at the nucleus as well. You can find that few nucleus are at this particular level and few nucleus are at this particular level. Nucleus at two different levels. Why? Due to the difference in sizes of the cell. This is pseudostratified ciliated simple columnar epithelium. Next. So we are done with simple epithelium. Now let's look at compound epithelium. What did I say? In compound epithelium, you will find nucleus present at different levels in multiple layers. So you can find lots of nucleus in one layer, nucleus in multiple layers. But then what you need to observe is are the cells at the top. The cells at the top, since it contains flat nucleus, the nucleus are present in multiple layer, stratified. Since the topmost cells, they are flat, it is stratified squamous epithelium. But then there's one more thing to observe: the presence or absence of keratin. You can see that at the top, keratin is not present. So it is non-keratinized stratified squamous epithelium. Okay, let's say keratin was present. It would look something like this. Again, the same features. You can find cells in multiple layers, but then the topmost cells, they are flat. Covering the epithelium, you can find this highly stained, branched, lamellated deposition of keratin. So if this highly stained, branched lamella of keratin is present on top of the epithelium, it is keratinized. But then if it is not present, then it is non-keratinized. So you have stratified squamous epithelium without keratin deposition. So non-keratinized stratified squamous epithelium. Then you have stratified squamous epithelium with keratin deposition. So this is keratinized stratified squamous epithelium. Next, you have stratified cuboidal epithelium. In stratified cuboidal epithelium, you need to observe for round nucleus arranged in multiple layers. So you have these round nucleus, but then these round nucleus, they are present in one layer. Surrounding this layer is the other layer. So there's multiple layers of round nucleus. So it has to be stratified cuboidal epithelium. Next up. So this one is a lot unique and easy to identify due to the shape of the cells. So you can observe that the bottom cells, you can see that the bottom cells, wait, okay, so the bottom cells, they are round, right? So at the bottom you have round cell. On top of that, this cell right here is pear shaped, so round cell. On top of that you have pear shaped cell, and then on top of that you can find dome shaped cell. So you have round cell, pear shaped cell, and then dome cell. Round cell, pear shaped cell, and then dome cell. These cells, they possess the ability to stretch. So this is your transitional epithelium. So these are all the epithelium that you need to be able to identify for your anatomy practicals. Let's look at all of them one last time. So how will you identify them? First, spaces are present. So it has to be either alveoli or Bowman's capsule. They are lined by simple squamous epithelium. To confirm, look at the cells. These cells, they are flat and arranged in a single layer. So this is simple squamous epithelium. Second, you can find these follicles. Lining these follicles, there are round cells. Round cells means cuboidal. So this is, round cells are arranged in single layer. So this is simple cuboidal epithelium. Next, so you can find that there are elongated cells present inside pillar-like cells, and the elongated nucleus present inside pillar-like cells, and these nucleus, they are arranged in a single layer. So this is simple columnar epithelium. Next, you can find that there are cilia, and nucleus, they are present at two different levels or multiple levels. So this is pseudostratified epithelium. Next, you can find that there are multiple nucleus arranged in multiple layers, and the nucleus at the topmost level, they are flat, and there is no deposition of keratin. So this is non-keratinized stratified squamous epithelium. Next, so there are these flat cells at the top, and there are cells arranged in multiple layers. So stratified squamous, since there is dense deposition of keratin, this is keratinized stratified squamous epithelium. Next, you can find that there are bunch of cuboidal cells. And these cuboidal cells, they are arranged in multiple layers. Since nucleus, they are present in multiple layers. Which type of nucleus? Round nucleus. So this is stratified cuboidal epithelium. Next, you can see that the cells that are present in multiple layers, and the cells at the bottom, they are round. Right on top of that, you have pear-shaped cell, and on top of that, there's dome-shaped cell. So this is transitional epithelium possessing the ability to stretch. So that's all for epithelial tissue. Next up, you are going to have connective tissue.